I hope that everyone has been enjoying the meeting as much as I have. This has been a tremendous display of fantastic science and exciting news for the progress of breast cancer. I am Luisa Arispe. I'm at Northwestern University, and I had the privilege this year to participate and chair the 2024 Award Selection Committee for the 7th recipient of the AACR Distinguished Leadership Lectureship in Breast Cancer Research. Since 2008, the AACR has been distributing this special award to recognize outstanding science that has inspired or has the potential to inspire new perspectives on etiology, diagnosis, treatment, or prevention of breast cancer. I would like also to take a moment to thank AFLAC, because since 2008, AFLAC has generously supported this special award, which continues to provide an important platform by which to recognize and honor quintessential leaders in the breast cancer research field. Please join me in thanking AFLAC for their ongoing support. This year's award recipient, Dr. Steffi Oesterreich, expertly embodies the purpose of this award to recognize and celebrate pioneering leaders in breast cancer research. Dr. Oesterreich is currently professor and vice chair of pharmacology and chemical biology at the University of Pittsburgh, as well as the director of education at the Women's Center Research Center. Dr. Oesterreich is being recognized for her pioneering research involving the investigation of the role of estrogen receptor-positive tumor cells in resistance to endocrine therapy, and for championing the concept that invasive lobular carcinoma should be diagnosed and treated differently from other forms of breast cancer. She is revered for being an international leader in translational breast cancer research, and especially in invasive lobular carcinoma of the breast. Her work has resulted in an international paradigm shift that involves the recognition of invasive lobular carcinoma as a distinct biological entity amongst breast cancer subtypes. Dr. Oesterreich's career spans 30 years. Initially, her research focused on the understanding of the biology of estrogen receptor alpha, and as she was part of the very first team credited with reporting the first naturally occurring ER mutations in breast cancer. Subsequently, in the year 2000, a number of publications, Dr. Oesterreich elucidated the role of estrogen receptor as a transcription factor and demonstrated comprehensively how ER not only activates gene expression, but equally represses gene expression. These studies offer crucial insight into how ER is able to regulate the breast cancer transcriptome and further hinted at ER's vital role in epigenetic reprogramming. In 2010, Dr. Oesterreich was recruited to the University of Pittsburgh, where she established her independent laboratory with the mission to further the understanding of the development and progression of breast cancer and to improve the clinical management of breast cancer patients with the ultimate goal of delivering better treatment and outcomes. Alongside her multidisciplinary team, she is currently investigating how estrogen receptor-positive breast cancers acquire resistance to endocrine therapies. Dr. Oesterreich is accomplishing this feat by employing systems biology approaches involving a combination of single-cell and bulk sequencing, as well as computational methods and biological models to identify and validate new drivers and therapeutic targets. More specifically, her efforts have and continue to be focused on characterizing hotspot estrogen receptor mutations present at invasive lobular carcinoma, or ILC, to guide the generation of more effective therapies for patients. Dr. Oesterreich has been a true pioneer in the study of this disease, which, as many of you know, is difficult to detect and commonly metastasizes to very unusual sites. She has shown that ILC exhibits variable patterns of nuclear receptor activity, metastatic spread, metabolism, and immune modulation, and presents with genetic drivers that differ from those commonly associated with invasive ductal breast cancer. Notably, Dr. Oesterreich has been essential to the establishment of a rapid autopsy program now called Hope for Others. This innovative program aims to bring together a large multidisciplinary team, including medical oncologists, pathologists, radiologists, translational researchers, bioinformaticians, and breast cancer advocates, with the shared goal of positively impacting patients' lives by guiding the generation of novel precision medicine approaches for ILC. This effort further illustrates how Dr. Oesterreich has been a vital champion for ILC advocacy. As the founding chair of the Scientific Advisory Board for the Lobular Breast Cancer Alliance Advocacy Group, she has played a pivotal role in international advocacy and research programs for ILC. And it has heralded for organizing the first ever international ILC Symposium in 2016. During her prolific career, Dr. Oesterreich has published over 200 peer-reviewed manuscripts in highly respected journals dedicated to many important research areas, ranging from molecular biology to translational and clinical studies. Consistent with her remarkable impact on breast cancer research, Dr. Oesterreich has received numerous awards and honors, including the Terry L. Chapman Award, the PNC Elise Hillman Distinguished Scholar Award, the Scheer Family Foundation Endowed Chair of Breast Cancer Research, and the Merrill J. Egerin Excellence in Scientific Leadership Award. Lastly, Dr. Oesterreich has proven to be an outstanding and selfless mentor for her trainees. She has enabled the success of many by consistently providing an enriching, team-driven environment that arms her trainees with the skills necessary to be able to establish successful scientific careers. Above all, countless women have and continue to receive breast cancer care because of Dr. Oesterreich's unparalleled and groundbreaking research. It is my great honor to introduce Dr. Oesterreich as this year's recipient of the AACR Distinguished Lectureship in Breast Cancer Research.
Thank you very much for this kind introduction. Thank you to AACR, the selection committee, and AFLAC. And looking at the list of prior awardees, I'm really deeply humbled and grateful to receive this award. And I hope somebody is going to set this timer back. This is not the real timer, right? Alrighty, I guess I need to speed up. Do I just push this here? No financial disclosures. I'll give you a few sentences of how I got here, the research areas we have been working on as examples in ILC, and then some unique aspects of ILC research, modeling, meetings, patient advocates, and collaborations. On the bottom of the slide, you actually see a picture of myself and my husband, Dr. Adrian Lee. We had independent breast cancer research careers for more than 20 years. And just a few years ago, we actually merged our labs. I have 2 slides on how I got here. So I actually grew up in East Berlin. I went to Humboldt University and Max Delbrück Center, and I was very excited about breast cancer research then. The wall came down, I was a graduate student, and I had the chance to meet Bill McGuire in Berlin at a cancer meeting. And I just grabbed him to my poster and said, I work on stress proteins, I want to work in your lab as a postdoc, is that okay? And he said, well, you need to come out to San Antonio and interview. That's what I did. After actually also driving Bill McGuire around the wall in East Berlin, little East German Trabant car, which he was very fascinated about. Anyway, then I went to San Antonio a year later. Somebody picked me up at the airport and said, unfortunately, Bill McGuire passed away 2 days earlier, which was a huge tragedy for his family, for his sons, for breast cancer research, for many, many patients, just because he was brilliant. And it really was horrible. But I have to say, I also thought a little bit what's going to happen to me. And I have to say, and it's a story I can share with people over a glass of wine or two. But the long story short, Dr. Ken Osborne took over the division, and I was very, very grateful for Ken's mentorship over the years. He was the first who really taught me translational research, because he attended most of the lab meetings from the trainees. He would always come in and say, you know, I don't care about your beautiful Western blot. I do care, but how does it help my patients? And we were always forced to really think about clinical relevance of what we did. I then walked in Suzanne Fuqua's lab on estrogen receptor. Suzanne at the time discovered ER mutation. It was a super exciting time and field, and it really was so stimulating. And upon move of the group to Baylor College of Medicine, I worked on ER cofactors. This was a great, great environment with Bert O'Malley being there, who, by the way, actually grew up and trained in Pittsburgh. And this was when I was exposed to invasive lobular breast cancer for the first time, because when I looked at ER cofactors, which I then worked on, and I cloned a few, looked in Oncomine at the time, the database, and they were higher on ILC, and I honestly had no idea what ILC was. I looked it up and then learned, yeah, so this is affects 10 to 15% of all breast cancers, mostly ER positive, in contrast to the more common invasive ductal cancer, now also called invasive breast cancer of no special type, NST, which again is the majority of breast cancer. ILC is primarily diagnosed based on H&E. The hallmark is loss of ecdyterin. As a result of this, it has this infiltrative growth of discohesive cells. One slide on the history. It was originally described by James Ewing, who also was born in Pittsburgh in 1992, a textbook on neoplastic diseases. But the term lobular carcinoma was actually coined in 1941 by Fudde and Stewart. So we then moved to Pittsburgh in 2010 to work there at the UPMC Hillman Cancer Center, which in many ways enabled my studies on lobular breast cancer. Dr. Nancy Davidson, this is here from a hike from Adrian, myself, her husband, Tom Kensler. And so Nancy was very excited about studying lobular breast cancer, and David Dubbs was at the time the chief of breast cancer pathology, and David had been working on ILC for many years. He was one of the first who really proposed to improve diagnosis of ILC, in part by using ecdyterin staining or by using a dual staining for ecdyterin P120, where you do co-staining, and as a result of loss of ecdyterin, you actually see cytoplasmic P120 staining. So this was a really, really fantastic start. Matt Sikora then joined the lab and worked on estrogen receptor action in lobular breast cancer. And here you can see Matt's studies on estrogen and anti-estrogen action in 2 cell lines. These were the workhorses of lobular breast cancer at the time. And without going into detail, he found that in MDA134, tamoxifen there was partial resistance and it functioned as an agonist. And in SAM44, there was a weaker estrogen response, and actually there was ligand-independent activity of the estrogen receptor. So we clearly had 2 models pointing towards different responses of ER. I was then very fortunate that Dr. Chuck Peru invited me to join the TCGA group, which was a first large molecular analysis of comparing ILCs and IDCs. And one of the most discriminating features between IDC and ILC were actually changes in ER cofactors. So there were more mutations in FOXA1 and there were fewer changes in GATA3, somewhat pointing towards differences in mechanisms to mediate ER transcriptional programs. And just as a side note, this study actually, for the first time, really large number and well done, was showing that the vast majority of lobular tumors had lost ecdyterin. So what about endocrine treatment if there are hints for differential activity of ER? Dr. Metzger-Filo had published a paper on suggesting that the relative effectiveness of letrozole compared to tamoxifen in patients is actually weaker, so more tamoxifen resistance. I thought it was a good idea to do this as a meta-analysis. I reached out to Dr. Metzger-Filo, to Dr. Mitch Dowsett and David Dabbs, and we contacted and then collaborated with the ABCTCG group analyzing ATAC, TEAM, and BIG198 response AI comparing tamoxifen. We had central pathology as well as equiterine staining. The data was shown a few years ago here at San Antonio. The manuscript is in preparation, and the final result is there was no significant difference in efficacy of AI versus tamoxifen comparing ILC versus NSD. But we also realized that there was really large discordance in ILC diagnosis, and there was overall worse outcome for patients with ILC, clearly suggesting we need biomarkers for response for hormonal treatment in patients with ILC. So one of the ways we did this was starting a preoperative window trial, an endocrine response in ILC. At the time, originally it was started with Dr. Jankowitz, and very grateful for Komen for supporting this. And upon Rachel's move to Penn, Dr. Priscilla McAuliffe became PI of this study. And I want to say she is presenting this tonight in the poster discussion session. So I'll be brief. The patients are exposed to fulvestrant, anastrozole, tamoxifen, and for 3 weeks before surgery. And again, this is a TBCR study. They're grateful to so many groups for hanging in there, doing this trial, completing this. Again, Priscilla will present this tonight. I just want to say that the analysis showed there was no difference in Ki-67 drop between anastrozole and tam. There was a significant difference between fulvestrant and tamoxifen. And please join the ILC poster discussion session tonight. What about outcome? There were a few studies showing that most likely outcome in the long run is worse for patients with ILC. We set out to do this with a large number of patients, with a limited number of institutions. We did this as part of the Great Lakes Breast Cancer ILC Consortium. There are 3 institutions, Pitt, UPMC, Case Western Cleveland Clinic, and OSU. We had more than 30,000 patients, of those 3,600 with ILC. And you can see on the slide that the curves really separate. For disease-free survival and overall survival after approximately 6 years, patients with ILC suffer from more late recurrences. More recently, this was confirmed in patients who were treated with chemotherapy. This was a fantastic collaboration with the NSCBP. The study was led by Julia Foley from the UPMC Hillman Cancer Center. And this was recently published, but basically the analysis showed, using clinical trial data from 4 NSCBP trials, in which patients were treated with chemotherapy, that again, patients with ILC have more late recurrences, shown on the bottom of the slide and on the right. So what options are there for therapy? I mean, in the end, I think one of the goals for all of us is really to have precision oncology, precision therapy for patients with ILC. There was a great study done on an immuno-oncology trial, the Gelato trial. It was published last year, and although negative, the study is really fantastic because there are great correlative science as part of this publication. I encourage you all to read this. So we set out to say, okay, we really want to do a comprehensive characterization of the immune microenvironment in invasive lobular breast cancer. This is a large collaborative study. And the first thing we found, and this was already done by shown by Christine Desmet and others previously, that overall the infiltration of immune cells in ILC, shown here in purple in the middle, compared to IDC is lower. However, there are tumors which are infiltrated with immune cells. And if you look at this in more detail, it is heavily dominated by infiltration of macrophages, both in the tumor as well as in the stroma region. And specifically, there's actually enhancement of M2-like macrophages in the ILCs. This is, again, I said this is a great collaboration with our immune colleagues at UPMC Hillman Cancer Center, especially Dario Vignali. And this was actually followed up by Chelsea Chen, a student, who then went back to look at some of the data and actually looked at gene expression patterns associated with macrophage infiltration and showed there are pathways specifically in the ECM which might predict an infiltration or not of macrophages into ILC tumors. What about other druggable pathways in lobular cancer? The TCGA study, which I mentioned earlier, showed that inactivation of PTEN was enhanced in lobular tumors, significantly more mutation. And you can see on this slide, on this heat map, which is a protein analysis, that overall the AKT PI3 kinase is significantly activated in lobular tumors compared to ductal tumors. We think this is due to two reasons. First, as a result of EKTRN, the cell membrane changes, the growth factor receptors are more active, and at the same time, there are increased growth factor levels in lobular tumors. Dr. Patrick Sturkson has done a couple of studies really showing this. We have shown a couple of studies as well showing this, and PI3 kinase, AKT, mTOR trials, we need more of those in patients for ILC. HER2 mutations, very interesting topic in lobular breast cancer. A couple of studies from Christine Desimet from Foundation Medicine showing they are enriched in lobular breast cancer. We did a study with Simon Johnson a few years ago, showed that there are more HER2 mutations in patients with ILC, and the patients actually do worse. We have a student in the lab, Jay, who's doing functional studies with this. He's using ILC models, patient-derived organoids with endogenous HER2 mutations or with CRISPR-engineered mutations, and again, without going through details, on the bottom left of the slide, you see that in those patient-derived organoids which have HER2 mutations, they have much more active phosphor-HER2, phosphor-HER3, phosphor-AKT, and phosphor-ERK signaling, and then these PDOs show increased sensitivity to neratinib as well as to the TDXD. This is an area which clearly deserves more studies in ILC. What about metastasis? We looked at metastatic sites in patients with ILC using two different databases. One was the UPMC McGee metastatic database on the top, and then in collaboration with Foundation Medicine. On the right, you see the number of patients. So these were significantly large studies for patients with metastatic IDC and ILC. Two results. First, there are decreased metastasis to visceral site, like liver and lung, and there are increased metastasis to the GI tract and the urogenital tract. Having the large UPMC database, we were also able to look for relatively rare sites of metastasis. So we actually we did a study showing that metastasis to the orbital and periorbital space are significantly enhanced. We are very interested in metastasis in ILC. We had a poster yesterday if anybody wants to talk more about this. This is for ovarian molecular analysis in ovarian metastasis. Very happy to chat about this. So the second part of the talk will focus more, I think, more unique aspects of ILC research. I will have 2 slides on modeling. I mentioned earlier that when we started the research 15 years ago, there were only 2 cell lines for ILC, which clearly isn't sufficient. So over the years, we realized that some of these cell lines might actually be lobular cell lines. They were just not characterized as such. Somebody else published on ILC-like cell lines. So we got all of those together, characterized DNA at the DNA, RNA, protein level, and then also started a collaboration with Abroad to really do RNAi CRISPR screens, to do drug sensitivity screens, and the data is available on bioRxiv. I should say that we also have an interest to work with our bioinformatician and biostatistician to do these congruence model, to really figure out which model represents which tumor. So this is also ongoing. What about PDOs? This is spearheaded by the Institute of Precision Medicine, which is run by Adrian Lee. Just here an example for 2 different PDOs on the left, an IDC on the right, an ILC PDO. Again, you can see when you do this co-stain of EKTRN, this P120, that the P120 in lobular tumors moves to the cytoplasm and clearly characterizes the patient-derived organoid. So without going into this in detail, we have a pretty large study ongoing that is run by Jack Moe and Huda. It's initiated by BCRF through support from Lee Pate, and it's a very extensive characterization of the organoids of the associated clinical samples with bulk as well as single-cell sequencing. And the goal is to have the PDOs, which I show you on the bottom right, and make them available to the larger research community. We also model ILC in vivo in genetically engineered models. There are great studies out there from a number of groups based on loss of EKTRN, PDOX model, and the MIND model. There are somewhat limited metastasis study out there, so we have labeled 2 of these cell lines. We can inject them in the memory fat bed. They go as primary tumor, they grow as metastasis, and they reflect metastasis which we see in patients, like, for example, the mice get metastasis to the bone and to the ovary. But what I think is most exciting in this model is actually they get leptomeningeal metastases, which are enriched in patients with ILC, and you can see this here on the right, and H&E stains, as well as when you take an antibody for human cytokeratin 19. This is actually, and some of these studies we are now continuing in collaboration with Sean Egan and Yi Li through the NCI Oncology Models Forum. So just a few slides at the end was about meetings, collaboration, and patient advocates. It was mentioned earlier, we had the first ILC meetings 2016 in Pittsburgh, where we got everybody together. This was super exciting. We now have a meeting almost every year here in the middle at different places. We had one just a few months ago run by Christine Desmond in Leuven. It was a great success. And the unique aspect of these meetings, what I would like to highlight, is that already at the first meeting, we had invited patients and patient advocates. And this is where I actually met Lee Pate. There's the red circle around her face here on the right side. She came to me and said, I was recently diagnosed with lobular breast cancer. I really would like to do more patient advocacy. And we met multiple times. We had phone calls. I chaired the scientific advisory board of the Lobular Breast Cancer Alliance. We had the first meetings here. We had a poster here. It was super exciting. Bugging San Antonio, we actually had a mini symposium a few years ago. LBCA has now grown. It's run by Laurie Hutchinson. It's now a 501c3 nonprofit organization. It partners with multiple organizations for ILC grants and disseminates ILC information and many more. The advocacy has grown. There are other national and international advocacy organizations. I list some of those here. We work together. There's a paper, this was actually spearheaded by Lee Pate at the time, to describe how researchers and patient advocates can work together. And there's also now an international group of patient advocates called GlobMob, who represent the different organizations and who help to organize the yearly ILC Symposium. And I want to end with 2 slides on interaction collaborations. So I think because the field is still relatively small, there are really active collaborations. There are many studies initiated by the ELBCC, spearheaded by Dr. Dirksen and Dr. Christine Desmet and Dr. Solomon. Just a few examples. They have done a tremendous job to move pathology forward, surveys, working groups, and then recommendation for pathology. There's interaction among the rapid autopsy teams from Pittsburgh, though from others, from KU Leuven, the Abteiler program, specifically for patients with ILC. In the models world, there are so many great collaborations, as well as in the Great Lakes Breast Cancer ILC Consortium. And one of the examples is really we have done a survey. Everybody worked together on this. The survey was responded by, we had more than 1,700 responses from more than 66 countries. The results are shown here on the right, but to summarize, there were differences between the different stakeholders in the priority areas, but the priority research areas are screening, diagnosis, imaging, dormancy, ILC modeling, treatment resistance, genomic predictors, and novel therapies and repurposing drugs, especially doing clinical trials for ILC. And this is a slide, my last data slide. Thinking more of clinical trials, Dr. Julia Foley recently opened a clinical trial called LBC Monitor. It's a liquid biopsy trial for patients with metastatic ILC. The idea is to de-intensify use for CDK4/6 in the first-line endocrine treatment setting. The ctDNA approaches is clearly specifically useful for patients with ILC given the imaging challenges. We have shown that the Signatera assay in 66 patients with metastatic ILC showed 90% baseline detection rate and concordance with clinical response, and therefore the trial was designed as follows. Patients with metastatic ILC have liquid biopsies, baseline 4 weeks, 8 weeks, 12 weeks, and then after that the physician choice treatment under estrogen minus plus CDK4/6 inhibitor. And the primary objective is to find ctDNA dynamics, and the secondary is concordance, molecular and radiographic response, and hopefully subsequently setting up a larger trial. So to summarize, primary and advanced ILC has unique molecular and clinical features. There is clearly increased awareness and interest of ILC as an independent breast cancer subtype by clinicians, researchers, and patients. An example is the poster spotlight session. I hope most of you go there tonight, and there are more than 50 posters on ILC at this year's San Antonio Breast Cancer Symposium. ILC models are becoming more available. Trials are ongoing for patients, but we clearly need more, and this is a priority area. Collaborations are absolutely critical given you have smaller number of patient populations. Some research priority for us specifically are in the diagnosis area, pathology, use of AI, ILC variants, liquid biopsy and genomic markers, imaging, especially using novel tracers, dormancy, coming up with clever models, as well as using the autopsy programs. And novel treatment targets. I mentioned growth factor receptor, PI3 kinase AKT, tumor microenvironment, ECM, immune cells, and more to be determined. And clearly, lineage of disease is an open question. My last slide, I do want to thank various people. I want to thank my family, especially my parents, who installed curiosity in science for me. My husband, Adrian. We have 2 daughters. They had 2 big events over the last 12 months. One got married, one graduated. And the mentors. I want to mention Nancy Davidson, I mentioned already, and Ken Osborne, who have been incredible mentors. I also want to mention Mark Lippman, who wasn't a direct mentor, but who I interacted each year multiple times and has given me most valuable advice throughout my career. There are many, many people from the lab, from the current lab, as well as lab alumni. I specifically mentioned the folks who worked on ILC topics here. Slide is too small to mention everybody. Tremendous collaborators throughout the years. And I want to end, my fierce friends, I have a photo here. Patients, patient advocates, and funding. And if I can have like 10 more seconds, I just want to mention specifically, again, thanks for all the funders. We couldn't do anything without the funding. I do want to mention BCRF because when I started the ILC research, I submitted multiple grants, one after the other. They weren't funded, and then I was very, very grateful to receive BCRF funding, which then really started our ILC research. And I want to mention Lee Pate again because she unfortunately passed way too early, and she was always so grateful for all the research, but I think in the end, I learned more from her than she learned from me. So, and thanks all of you for listening to me.